Okay, this lecture is to provide you with information about transport across the membrane. These are the following outcomes that will be covered. So transport across the membrane, the transport part gives the membrane what we call selective permeability, which means the ability of the membrane to control what crosses. We've talked about the concept of semi-permeable, which is just based on the characteristics of a phospholipid membrane. So certain molecules can just move across, but most molecules can't. And so we're going to talk about how do we move those molecules that don't go across just by simple diffusion. So the three main ways solutes can move across a membrane is simple diffusion, and that means a molecule is going right through the membrane. Or the phospholipid bilayer. Next, we have facilitated diffusion, which means that a protein is required for movement of the molecule. Across the membrane. Let's see if we can fit that. Okay. So these, the key word is diffusion. So whether it's simple diffusion or facilitated diffusion, it means that this is what we call passive transport. No energy required. And the molecules are moving down their concentration gradient. So it says down here, passive transport in the direction of the electrochemical gradient. Molecules move down their concentration gradient, which means the molecules are moving from high to low concentration. So over on this side of the phospholipid bilayer, it's telling you that whatever we're trying to move, this little circle, has a high concentration on the top of the membrane and a low concentration on the bottom. So simple diffusion, facilitated diffusion, whether it's with a channel protein or a carrier protein, allows for molecules to move across the membrane from high to low concentration. So let's look at passive transport again. Okay. Here again are some examples. I like this diagram better for the transport or the carrier protein. So we have molecules that are going through by simple diffusion. We have molecules that are going through a channel protein. So channel-mediated facilitated diffusion. So high to low concentration still. But the molecules need some help. Remember, a channel is like a tunnel through the membrane. But I want you to know too that this is still selective. So not just anything can go through a channel protein. There are size constraints, there are molecules inside that make up the channel protein called amino acids that are going to regulate. So it's still regulate what crosses. So it's still selective. But things can move faster if they're the right molecule. Whereas with carrier-mediated facilitated diffusion, what I want you to see is as the reaction is happening or the molecule is being moved across, the carrier protein changes shape. Okay. So it grabs a molecule on this side of the membrane, changes shape, and releases the molecule on the other side of the membrane. Okay. This is still going from high to low, so this is still passive transport, and we use the term diffusion, and diffusion is used for solutes. There is a different video, remember this is concentration, those little brackets. There is a different video that talks about water and osmosis. And so when water moves from high water to low water concentration, we call that osmosis. So gases like CO2 and oxygen, very small uncharged molecules such as ethanol, moderately permeable, so water, urea. These are also small polar molecules, so they have a slight charge. Those can go through by simple diffusion. The other molecules over on the bottom of our membrane that cannot cross, like sugars, ions, charged polar molecules, so larger macromolecules, things like that, cannot pass through. So they're going to have to go through by facilitated diffusion. Oh, and there's one we forgot up here, which is hydrophobic molecules can also fit through the membrane. So any type of diffusion you have, whether it's simple or facilitated, means there's no energy required. You're moving from high to low concentration. And you see on the bottom there are links to videos that help explain this as well. Active transport is different. Active transport means it requires energy, ATP. Excuse me. It also requires some kind of transporter protein, so a carrier or transporter protein. And now we call this protein a pump, because what the protein is doing is it's pumping molecules against its concentration gradient. That means the molecules are moving from low to high. Okay, so this is harder to do. This requires energy. Now you don't have to know which molecules move which way, by active transport, by a channel for facilitated diffusion, or by a carrier for facilitated diffusion. But you should be able to interpret diagrams, or given a kind of story problem, be able to draw the type of transport for that molecule. So just to reiterate, we have different protein types. We have channels, which are like a tunnel. We have transporter or carrier proteins, and we have pumps. I like this diagram as well because it's trying to illustrate that a carrier protein is going to physically change shape in order to move the molecule. And you'll notice in some of these we talk about a solute. So a solute is all the stuff dissolved in water. So remember, we're mostly water. You have water on the outside of your cells, you have water on the inside. So whatever is dissolved in the water, like sugars, ions, whatever is dissolved there, that's the solute. And so that's what the protein channels, transporters, and pumps are moving. They're moving the solute. Again, there's another separate video that talks about the movement of water. Now you can kind of put these concepts together, and what the cells do a lot is they do what's called primary and secondary active transport. And this name can be a little bit confusing. So I want you to see that on the left here is the primary active transport. So primary active transport requires ATP energy. Okay, that's right here. So in order to move these ions, we need energy. And it's usually because, as you can see, we're moving ions against the concentration gradient. So there's a low amount of sodium here to the high amount of sodium on the outside, and it moves things just the opposite as well. This is a sodium-potassium pump. We'll talk more about that in a minute. Moves potassium ions from low to high concentration. The point of this is that the cell, so primary active transport, let's erase this, makes a concentration gradient on one side of the membrane. So what's happening here is the cell is pumping sodium out, and sodium is becoming high in concentration, so there's lots of sodium out here. And so sodium is going to come back into the cell down its concentration gradient through a second protein. So active, primary and secondary active transport have two totally different proteins that are being used. The first protein requires ATP, and you are pumping things against the gradient. You make a concentration gradient, and now sodium can enter. Sodium enters the cell by facilitated diffusion. Okay, so facilitated diffusion again is moving a solute or a molecule with its concentration gradient from high to low, and in doing so, it can also bring in a second molecule. And I'm going to show you one more example of this. What I want you to get from this is that the cell is on purpose creating a concentration gradient, and then using that concentration gradient to bring in a second molecule. Let's look at another example of this. Excuse me. So here is our primary active transport. In this case, the cell is pumping H plus out of the cell. So here's the cytosol, so this is outside. Okay, so it's using active transport. Makes a high concentration of H plus on the outside, and then uses the concentration gradient, so the high level of H plus, to bring a second molecule in this case sucrose, without more energy. So secondary active transport alone does not require energy. Primary requires energy, and I usually write energy just as a little funky E. So the first molecule is moved by active transport. It creates this concentration gradient. It moves back down its concentration gradient and brings in a second molecule with it. So over here we have a pump. Over here we have a carrier. Over here we have facilitated diffusion. Of the hydrogen ions. And so we call the movement of sucrose, as secondary active transport. A couple other terms you can use to describe transport across a membrane is what or how many types of molecules and in what direction. So Symport means two types of molecules are moving together in the same direction. Both are coming, in this case, into the cell. Antiport, you have two types of molecules moving in opposite directions. So one is coming in, one is going out. And uniport is just one molecule and obviously one direction. Now, this same protein, maybe it's a protein channel, might allow the molecule to move back out, but it's one molecule in one direction at a time. So if you took a look at this is another illustration of the sodium-potassium pump. This is a super important pump to learn about because it's key at making concentration gradients. It's very important in nerve cells. And if you looked and described this, you would say, okay, well, this is active transport. Right? And this is also an example of antiport, because the sodium is moving out of the cell and the potassium is moving in. So you have two different types of molecules moving in opposite directions. So the next few slides I want to show you are just some examples. These are not things you need to memorize, but these are examples of transporting, and you might see an example on an exam or a quiz, and I might ask you to describe that type of transport. So for instance, you have amino acids and sodium ions coming in, doesn't show any ATP, so this is facilitated diffusion. You don't see ATP, it's got to be diffusion. Facilitated because this represents a protein, and it's Symport. Two types of molecules going in the same direction. Then that amino acid is moved by uniport, and here is, and what they should draw in here is a protein. Here is the sodium-potassium pump, okay, which is an example of active transport. And antiport. So you can see lots of Symport going on. Here's a uniport. Here again is our classic antiport. Here's what's happening in your kidney. Look at all these different molecules transporting across. Again, you don't have to memorize any of this. You just have to be able to interpret. So wherever you see ATP, you're going to know that's active transport. If there's no ATP, it's some kind of diffusion. If it has a protein, it's facilitated diffusion. Here, there's a good one. Well, they're blocking out water. I didn't see simple. They don't have simple diffusion. Here they're showing you that water doesn't really cross the membrane except through facilitated osmosis. And again, there's a different lecture to look at that. So, don't memorize any of this, just be able to interpret it. Excuse me. Here's one more example. Erythrocyte is your red blood cells. Here you've got carbon dioxide and oxygen and water moving through simple diffusion. You have carrier proteins helping with glucose transport and carbonate and chloride anion exchange. Here you have active transport with ATP, and also water moving in with an aquaporin. So water alone, simple diffusion, aquaporin, facilitated diffusion, facilitated osmosis is more accurate. So make yourself familiar with the different types of transports, the differences between simple diffusion, facilitated diffusion, and active transport, understand primary and secondary active transport. I like to give you lots of figures just because hopefully one of them makes sense to you and you can see how things are working. Let's just, last thing I want to do is make sure we covered all the objectives. So we described 3 ways in which solutes can move across the membrane. We talked about passive facilitated diffusion, and active transport. We talked about primary and secondary active transport. This is a really challenging concept for a lot of students, so make sure you look at that. Compare and contrast how channels, transporters, and pumps work. So go look between the difference between a tunnel and a transporter or a carrier protein. And here we're talking about facilitated diffusion and a pump. I really like to give compare and contrast questions for short answers, so definitely work on those, thinking about differences, thinking about similarities. Here, this is why I go back through here. Here's a topic that we didn't quite cover. Carrier and transport proteins can become saturated. Saturated, again, means you're at maximum capacity. So let's just go look at protein. Okay, so if I get rid of some of this squiggly. Okay, if you have a channel protein, right, things can just continuously go through as long as they're the right molecule and they fit. Carrier or transporter proteins have to grab something, make a shape change, and then release it. And grab something and make a shape change and release it. So they can become saturated, where there might be hundreds of molecules out here, but they're working as fast as they can. That affects the rate of the movement of the molecule across the membrane. So, carriers, transport proteins, and pumps can become saturated. They can reach maximum capacity, and even though there's more solute out there, they just can't continue to pump it in any faster. Versus a channel protein that, because of its structure, it can just continuously allow flow through it. We talked a little bit about the sodium-potassium pump and how it works. Let's just look at it one more time. Okay. What's happening with the sodium-potassium pump is things are only going one way for each molecule. So 3 sodium bind to this molecule, this protein, sorry. So the brown protein is the same. It's showing you a continuous, how the protein works in a continuous manner, as we don't have an animation. So it binds 3 sodium ions. It changes its shape, releases the 3 sodium ions, and now 2 potassium ions can come in and bind. The protein again changes shape and releases the potassium to the inside of the cell. So the sodium-potassium pump works by moving sodium, moving potassium, requiring ATP, and the protein itself will continuously change shape as it's moving the ions across the membrane. Okay, compare and contrast. So we talked about symport, antiport, and uniport. And then I gave you some examples of how you could look at an image and identify if it was a symporter, an antiporter, or a uniporter, or I could give you a description and you could draw it. All right, I hope that helps you with transport across a membrane.